Welcome to our deep dive into the tumor microenvironment, often abbreviated as TME. This complex ecosystem surrounds and interacts with tumor cells, playing a crucial role in cancer development, progression, and response to therapy. Understanding TME is essential for unraveling the mysteries of cancer biology and improving treatment strategies. In this video, we'll explore the key components, functions, and significance of the TME, setting the stage for a journey through cellular landscapes and molecular interactions. Let's begin by defining what the tumor microenvironment is and why it matters in the context of cancer research. Tumor microenvironment refers to the surrounding cellular and non-cellular components that interact with tumor cells. It includes immune cells, blood vessels, fibroblasts, signaling molecules, and the extracellular matrix. These elements collectively influence tumor growth, metastasis, and response to treatments. By understanding the TME, researchers can identify new targets for therapy and develop more effective cancer treatment. The TME is not static. It evolves as the tumor progresses, adapting to changes and influencing the behavior of cancer cells. Within the TME, a variety of cells play distinct roles. Immune cells such as T cells, macrophages, and dendritic cells can either attack tumor cells or paradoxically help them evade immune detection. Fibroblasts contribute to the structural framework and secrete factors that influence tumor growth. Endothelial cells form blood vessels, supplying nutrients and oxygen to the tumor. Each cell type interacts dynamically, creating a constantly shifting environment that impacts cancer progression. Non-cellular components of the TME are equally important. The extracellular matrix provides structural support and influences cell behavior. Signaling molecules, such as cytokines and growth factors, mediate communication between cells. These elements create a biochemical landscape that can either promote or inhibit tumor growth. Understanding these non-cellular factors is vital for developing therapies that target the TME and disrupt cancer-supporting processes. Immune cells within the TME can have dual roles. Some, like cytotoxic T cells, attack and destroy cancer cells. Others, such as regulatory T cells and certain macrophages, suppress immune responses, allowing tumors to grow unchecked. This balance between immune activation and suppression is a key factor in cancer progression. Therapies that modulate immune cell activity are at the forefront of cancer treatment, aiming to tip the balance in favor of tumor destruction. Fibroblasts are connective tissue cells that play a significant role in the TME. They produce extracellular matrix components and secrete growth factors that influence tumor behavior. Cancer-associated fibroblasts can promote tumor growth, invasion, and resistance to therapy. Targeting fibroblast activity is an emerging strategy in cancer treatment, aiming to disrupt the supportive environment that tumors rely on. Blood vessels within the TME are formed through a process called angiogenesis. Endothelial cells create new vessels to supply the tumor with nutrients and oxygen. This vascular network is essential for tumor growth and survival. However, abnormal blood vessel formation can lead to inefficient delivery of therapies and contribute to metastasis. Anti-angiogenic therapies aim to disrupt this process, starving tumors of their lifeline. Extracellular matrix (ECM) is a dynamic structure within the TME. It provides physical support and regulates cell movement, signaling, and growth. Changes in the ECM can facilitate tumor invasion and metastasis. Enzymes that remodel the ECM, such as matrix metalloproteinases, are often upregulated in cancer. Targeting ECM dynamics is a promising approach for limiting tumor spread. Signaling molecules within the TME, such as cytokines, chemokines, and growth factors, orchestrate communication between cells. These molecules can stimulate tumor growth, recruit immune cells, or promote inflammation. The balance of signaling molecules determines whether the TME supports or inhibits cancer progression. Therapies that target these signals are being developed to disrupt cancer-promoting pathways. Hypoxia, or low oxygen levels, is a common feature of the TME. As tumors grow, they outpace their blood supply, leading to oxygen deprivation. Hypoxia triggers adaptive responses in cancer cells, making them more aggressive and resistant to therapy. Understanding and targeting hypoxia is crucial for improving treatment outcomes and preventing tumor progression. Tumor cells within the TME undergo metabolic changes to survive and thrive. They often switch to glycolysis, a process that generates energy without oxygen. This metabolic shift supports rapid growth and creates a hostile environment for normal cells. Targeting cancer metabolism is an innovative approach to disrupt tumor survival mechanisms. Inflammation is a hallmark of the TME. Chronic inflammatory signals can promote tumor growth, angiogenesis, and immune suppression. The interplay between inflammatory cells and cancer cells creates a feedback loop that sustains tumor development. Anti-inflammatory therapies are being explored to break this cycle and restore immune balance. Tumors develop strategies to evade immune detection within the TME. They may express proteins that inhibit immune cell activity or recruit cells that suppress immune responses. This immune evasion allows cancer cells to survive and proliferate. Understanding these mechanisms is key to designing therapies that restore immune surveillance. Macrophages within the TME can adopt different roles. Tumor-associated macrophages, TAMs, often promote tumor growth, suppress immune responses, and facilitate metastasis. Targeting TAMs is a promising strategy to shift the TME towards anti-tumor activity and improve therapeutic outcomes. Cancer-associated fibroblasts, CAFs, are specialized cells within the TME. They secrete factors that remodel the extracellular matrix, promote tumor invasion, and support cancer cell survival. Targeting CAFs is an emerging area of research with the goal of disrupting the supportive niche that tumors depend on. Endothelial cells line blood vessels and are crucial for angiogenesis in the TME. They respond to signals from tumor cells, forming new vessels that supply nutrients and facilitate metastasis. Targeting endothelial cell function is a key strategy in anti-angiogenic therapies, aiming to cut off the tumor's supply lines. Remodeling of the extracellular matrix is a dynamic process in the TME. Enzymes such as matrix metalloproteinases break down ECM components, facilitating tumor invasion and metastasis. Inhibiting these enzymes is a potential therapeutic approach to limit cancer spread. The stroma is the supportive tissue surrounding tumor cells in the TME. It includes fibroblasts, immune cells, blood vessels, and ECM. The stroma provides structural support and influences tumor behavior. Targeting stromal components is a promising strategy for disrupting the tumor microenvironment. Cancer cells communicate with their surroundings through direct contact and signaling molecules. This communication shapes the TME, influencing immune responses, angiogenesis, and metastasis. Disrupting cancer cell communication is a potential avenue for therapy. The TME plays a critical role in metastasis, the spread of cancer to distant sites. Factors within the TME facilitate cancer cell migration, invasion, and survival in new environments. Understanding these processes is essential for developing therapies that prevent metastasis. The TME can contribute to resistance against cancer therapies. Factors such as hypoxia, ECM remodeling, and immune suppression create barriers to effective treatment. Overcoming therapy resistance requires strategies that target both cancer cells and their microenvironment. Immunotherapy harnesses the power of the immune system to fight cancer. The TME can influence the success of immunotherapy by modulating immune cell activity. Strategies to enhance immunotherapy include targeting immune suppressive cells and signaling pathways within the TME. Targeting the TME is an emerging approach in cancer therapy. By disrupting the supportive environment, therapies can enhance the effectiveness of traditional treatments and prevent tumor progression. This multifaceted strategy involves targeting cells, ECM, and signaling molecules. Personalized medicine tailors cancer treatment to individual patients based on their TME characteristics. By analyzing the cellular and molecular makeup of the TME, clinicians can select therapies that are most likely to succeed. This approach represents the future of cancer care. Biomarkers within the TME can predict treatment response and disease progression. These include proteins, genes, and signaling molecules that reflect the state of the microenvironment. Identifying reliable biomarkers is crucial for guiding therapy decisions. The TME presents challenges for drug delivery. Dense ECM, abnormal blood vessels, and high interstitial pressure can hinder the penetration of therapies. Innovative delivery systems are being developed to overcome these barriers and improve treatment efficacy. Tumors are heterogeneous, meaning they contain diverse cell populations and microenvironments. This heterogeneity complicates treatment and contributes to therapy resistance. Understanding the diversity within the TME is essential for developing effective therapies. The TME evolves over time as the tumor progresses. Changes in cell populations, ECM composition, and signaling pathways can alter the microenvironment's influence on cancer. Tracking these changes is important for adapting treatment strategies. Some cancer cells enter a dormant state within the TME, evading detection and surviving therapy. Dormant cells can later reactivate, leading to relapse. Understanding the mechanisms of dormancy is crucial for preventing recurrence. Immune checkpoints are proteins that regulate immune cell activity within the TME. Tumors exploit these checkpoints to suppress immune responses. Therapies that block immune checkpoints such as PD-1 and CTLA-4 inhibitors are revolutionizing cancer treatment. The metabolic landscape of the TME influences cancer cell survival and growth. Interactions between cancer cells and stromal cells can alter nutrient availability and metabolic pathways. Targeting metabolic interactions is a promising area of research. The TME can influence the effectiveness of various therapies, including chemotherapy, radiation, and immunotherapy. Factors such as hypoxia, ECM density, and immune suppression create obstacles to successful treatment. Overcoming these challenges requires a comprehensive understanding of the TME. The characteristics of the TME can influence cancer prognosis. Factors such as immune cell infiltration, ECM composition, and angiogenesis are associated with outcomes. Assessing the TME provides valuable information for predicting disease progression. The TME can contribute to cancer recurrence after treatment. Dormant cells, therapy resistance, and immune suppression create conditions for relapse. Strategies to modify the TME are being developed to reduce the risk of recurrence. The TME creates a metastatic niche that supports cancer cell survival in distant organs. Factors such as ECM remodeling, immune suppression, and angiogenesis facilitate metastasis. Targeting the metastatic niche is a key strategy for preventing cancer spread. Cancer stem cells within the TME are responsible for tumor initiation, progression, and recurrence. The microenvironment provides signals that maintain stem cell properties. Targeting cancer stem cells and their niche is a promising approach for durable cancer control. Immunosuppression within the TME is a major barrier to effective cancer therapy. Regulatory cells, signaling molecules, and metabolic changes suppress immune responses. Overcoming immunosuppression is essential for successful immunotherapy. The TME facilitates cancer cell invasion into surrounding tissues. ECM remodeling, signaling molecules, and cell interactions create pathways for invasion. Targeting these processes can limit tumor spread and improve outcomes. The angiogenic switch is a process where the TME promotes blood vessel formation. This switch is triggered by signaling molecules and hypoxia. Targeting the angiogenic switch is a strategy for controlling tumor growth. The TME modulates immune responses through cell interactions and signaling pathways. This modulation can either enhance or suppress anti-tumor activity. Therapies that restore immune balance are being developed to improve cancer outcomes. Drug resistance is a major challenge in cancer therapy. The TME contributes to resistance through hypoxia, ECM barriers, and immune suppression. Strategies to overcome drug resistance focus on modifying the microenvironment. The TME drives tumor progression through cell interactions, signaling, and structural changes. Understanding these processes is essential for developing therapies that halt cancer growth and spread. Clinical trials are testing therapies that target the TME. These trials evaluate the effectiveness of drugs that modify immune responses, ECM dynamics, and angiogenesis. Results from these trials will shape the future of cancer treatment. Research into the TME is rapidly advancing. New technologies such as single-cell analysis and spatial profiling are providing deeper insights into microenvironment dynamics. These advances will lead to more precise and effective cancer therapies. Summary: the tumor microenvironment is a complex and dynamic ecosystem that shapes cancer development, progression, and response to therapy. By understanding and targeting the TME, researchers and clinicians can develop more effective treatments and improve patient outcomes. The future of cancer therapy lies in unraveling the secrets of the microenvironment and harnessing its power for better health.